So in the 1960s, Francis Crick, who I mentioned earlier, he was involved with determining the double-stranded structure of DNA, won the Nobel Prize in 1953. Sidney Brenner won the Nobel Prize twice. I can't remember what years, but he was a very prolific scientist. They worked on figuring out how an mRNA instructs the ribosome to build a protein. And they were able to figure out that in the mRNA, every 3 adjacent nucleotides would signal to the ribosome as to a specific amino acid to add to the protein. And the 3 nucleotides, the group of 3, is called a codon. So each codon indicates 1 amino acid. And so the correlation of the codons, here you see the groups of 3 nucleotides, and then which amino acid they correspond to is shown here. And this chart is called the genetic code. So each group of 3 nucleotides, so like UUU, right here, corresponds to the addition of the phenylalanine. PHE is phenylalanine. This is leucine. This is isoleucine, methionine. You don't have to memorize these, but these 3 letters indicate the amino acid names. They're just abbreviations for the amino acid names. So depending on what the order of these little groups of 3 nucleotides occurs in the mRNA, that determines the order of certain amino acids being joined together to make the protein. There is one codon, AUG. That codon is the one that signals where the protein building will begin. It's called the start codon. And there are 3 codons that don't correspond to any amino acid, and those are the stop translation codons. So the start translation codon actually does cause a methionine, MET, to be added into the protein, but the stop codon does not indicate any amino acid to be added. So if you had an mRNA that had 100 codons, including 1 start codon and 1 stop codon, then you would end up with a protein that had 99 amino acids, because the 100th codon does not add an amino acid. So you'd only end up with 99 amino acids in that example, in that protein. However, proteins can be any length. They can be 100 amino acids long, or they can be 1,000, or they can be 5,000. It just depends on how long the mRNA is, which means how long the gene was. So there's no rule for length. All right, so tRNAs, T stands for transfer. Transfer RNAs are helper molecules, and what they do is they carry amino acids to the ribosome. The tRNA has a section, a 3 nucleotide section called the anticodon. The anticodon is in the tRNA, has 3 nucleotides that would be complementary to one of the codons in the mRNA. And then attached to that tRNA would be a certain kind of amino acid. So it gets a little complicated, but here's a space-filling model of a tRNA. At this end, there are 3 nucleotides. The whole thing is made of nucleotides, but these 3 nucleotides are special. They're in a particular position. And in this example, it's AAG. So that anticodon would be attracted to, in the mRNA, here's the mRNA, would be attracted to a UUC, for example. And so then UUC in the mRNA is associated with, this tRNA would carry a phenylalanine. So let me go back one slide. The UUC is in the mRNA. So do you see UUC indicates phenylalanine? So the genetic code is written from the mRNA point of view, not the tRNA point of view. The codons are in the mRNA, but there is a complementary anticodon in the tRNA, and that tRNA will always bring in a certain amino acid, in this case phenylalanine. You do not need to memorize the codons, the anticodons, and which amino acids they are associated with, because if I need you to answer a question about that, say on the test, for example, I would provide for you the genetic code. You would have that. So the mRNA has these codons. So for example, here you have A, U, G. That would be the start codon. And then you have these groups of 3, whatever they are. And then at the very end, it would be one of the stop codons, UAA, for example. So the mRNA has these codons in it. The ribosome is the thing that's going to somehow, we say, read or take the information from the mRNA, those codons, and use it to build a certain kind of protein. The ribosome comes in two parts. In the cytosol, it's in 2 pieces. We call them the large subunit and the small subunit. They're made up of protein and other kinds of RNAs. Not mRNAs, but other kinds of RNAs. We call those ribosomal RNAs. But those proteins and ribosomal RNAs bunch together and make this large subunit or this small subunit, and those are just sitting there in the cytosol. And when an mRNA comes out of the nucleus after transcription, these 2 pieces come together surrounding this mRNA. And I usually refer to it as like a hamburger bun. You have the lid, the large subunit is like the lid of the bun, and the small subunit is like the bottom of the bun. And then the mRNA is like a piece of bacon in the middle. Whatever you want to call it. But that's kind of how it happens. So the mRNA comes out of the nucleus, and the 2 parts of the ribosome kind of clamp on around it. If we sliced into the inside of the ribosome, we would see some little hollow spaces. And there's 3 hollow spaces. One's called the E site, one's called the P site, and one's called the A site. All right. So the amino acids, you may remember, have 2 functional groups. They all have an amino group and a carboxyl group. And they all have an alpha carbon and a hydrogen. And then they have what's called the R, or variable chain, or side chain. And so in this picture, the variable chains are shown in blue. And there's 20 amino acids commonly found in proteins on Earth. And they all have names. A lot of the names end with the letters INE. There's a few exceptions to that rule. Aspartate does not end in an INE. Glutamate does not end in INE. Tryptophan does not end in INE. The rest of them, the rest of them do. So these are the full-length names. Again, you don't need to memorize them. If you take biochemistry, you will not only get to memorize the name, you will also memorize the molecular structure, but that's in a future class, so don't worry about that now. But these amino acids have different R groups, different side chains, and so it means that depending on how and which amino acids are hooked together, it gives the final protein product a final shape and a final chemistry that's unique. And remember how a peptide bond is formed. It's formed between the hydroxyl end, actually part of the carboxyl, the OH part of the carboxyl of one amino acid, and a hydrogen, which is part of the amino group of another amino acid. And when that dehydration synthesis happens, you get the peptide bond, which is directly between that carbon and that nitrogen. So that's what the ribosome is doing. The ribosome itself is the enzyme that makes that peptide bond. So what happens is, and you can kind of see the ribosome in the background. This is happening in the ribosome, but what happens is, here's the mRNA here, and they're showing the start codon AUG here. So starting from the beginning, there's an amino acid that corresponds to AUG. It's called methionine, MET. The tRNA that would be carrying methionine would always have the anticodon UAC. And so when the AUG, and it has to be in that P site, remember the 3 places inside the ribosome? There's one called P. When the AUG is in that P site, then the tRNA that is complementary to AUG is UAC, comes into the P site and sticks there briefly, and it always brings methionine with it, so that brings a methionine into the ribosome. And then the ribosome grabs that methionine, and so when the tRNA leaves through the E site, E is exit, it doesn't have that methionine attached to it anymore because the methionine is left behind. The ribosome grabs it. The next codon is UUC, and we've already talked about that one a little bit. A tRNA with an AAG would be attracted to that one, and that carries phenylalanine. And so it moves in, and then the ribosome takes the methionine that was already there, and then now the phenylalanine that's just come in, and it makes the peptide bond between them. And then the ribosome is going to move forward. The ribosome is going to move in this direction. And so as it slides down, a new codon comes in, CGA for example, and there will be a tRNA that's got an anticodon that matches that, and it'll bring in the next amino acid and so on. And so you'll end up with methionine will always be the first amino acid in the chain, and then whatever's encoded next, in this case phenylalanine, and then whatever's next, in this case arginine, and so on and so forth. Eventually there will be a stop codon, so for example UAA is a stop codon. Eventually when a stop codon comes in, then the protein is done and everything is released. So that's how the right kind of protein is made. What happens if there's a mutation in the gene? Well, a mutation in the gene means in the DNA, in the chromosome, there would be a change in a nucleotide. So maybe an A gets changed to a G, or a C gets changed to a T, or anything can be possible. What causes mutations, a lot of what causes mutations in cells are just mistakes. DNA polymerase makes mistakes during DNA replication. And if the mistakes happen during DNA replication and they don't get fixed, then they become permanent. There can be also chemicals that come into the cell that cause changes to the DNA. Those are called mutagens. And also radiation is a mutagen, so UV light, X-rays. But whatever the cause of the change in the DNA, if there's a change, it can, if the change happens within a gene, it can change a codon. And so a base substitution would be just a term that describes a DNA mutation in which one base, like adenine, is substituted for another base like guanine, or C or T. So this is at the DNA level. But of course, if there's a mistake or a change at the DNA level, remember, the RNA polymerase during transcription is just copying the gene, and so it just copies whatever's there. It doesn't know that it's not supposed to be an A. It doesn't know what's supposed to be there. It just looks at what's there and makes a copy in the complementary mRNA. And so it can change what should be a certain codon. Maybe there's a UUC, which codes for phenylalanine, and maybe it gets changed to an AUC, which codes for something else. So it can cause a change in the protein product. And so we have different terms that describe the effect on the protein. If there's a base substitution mutation which changes an amino acid, like I said, a phenylalanine because it gets switched for something else because the codon is changed, that's called a missense mutation. All right? And so a missense mutation means the amino acid that gets inserted into the protein is not the one it's supposed to be in the normal copy of the gene. But you still end up with a full-length protein. There's just one amino acid that's wrong, so to speak. A silent mutation is when you have something like, let's say a codon that's UUU gets changed to UUC. Well, both of them indicate phenylalanine, so that would not be a change in the protein. So that would be called silent. There is a change at the DNA level, but there's no change in the protein level. A nonsense mutation is where you have a normal codon for an amino acid. Let's say you have a UUC. Let's do a UU. Now let me pick one. UAC gets changed to UAA. So UAC encodes an amino acid, but UAA is a stop codon. So if the mutation creates a stop codon where there's not supposed to be a stop codon, that kind of mutation is called a nonsense mutation. So you have to memorize these: silent, missense, and nonsense. All three of these are types. They are really describing the results of certain types of base substitutions. The other kind of basic kind of mutation you could have is called a frame shift. A frame shift is when there's an additional nucleotide accidentally inserted into a gene or removed. And what happens there, let's say that the normal, and this is not the whole protein, they're just kind of zooming in on part of it. Of course, all proteins start with AUG on the RNA. But let's say within the protein you have AGC, which indicates serine, GTA, which indicates valine. Let's say this is the normal gene. This is the normal protein product of that gene. Then, and they're showing the gene, not the RNA, by the way, but it doesn't matter. And then let's say that you have, they're saying, let's say that there's a deletion of this T and this A. So what would happen is now this G here gets grouped with the first two C's over here. So there's a regrouping. Do you see that? So serine is the same, but where the deletion happened, you get a change. So valine is not going to be added this time. Now it's going to be GCC, which is alanine. And then the C gets recombined with a T and an A, and that's leucine, and so on. So all the groupings after the deletion or insertion will be wrong compared to what it's supposed to be in the normal gene. And so you would end up with a protein that's just got a whole bunch of amino acids that aren't supposed to be what are in that protein. So that's called a frame shift, because when you delete one or more nucleotides, the regrouping, the codons just group the way, the cell doesn't know. It just groups in threes. So it's going to read the instructions incorrectly. And finally, I just want to remind you how this all works together with protein secretion, which you already know about. All right? So the gene is copied in the nucleus. The two strands are separated and mRNA is made from the gene. The transcription starts at the promoter, ends at the terminator. The mRNA is processed and comes out of the nucleus. All right? Then that mRNA gets sandwiched between two parts of a ribosome. And the translation begins. And what's sometimes going to happen is those first amino acids, the methionine and then whatever's next and whatever's next, start to hang out of the ribosome a little bit as the ribosome is moving down the mRNA and making that protein, which is getting longer and longer. The very first part of the protein, if the protein needs to be secreted, that very first part of the protein has a certain amino acid sequence called the signal sequence. And there's another protein called signal recognition particle that will take that ribosome and mRNA and the first part of the protein and stick it onto the rough ER. And then the rough ER will, the protein will thread into the rough ER and be pushed into the lumen of the rough ER and then be released. So the protein starts to be built just on a free ribosome floating in the cytosol. But if it's a protein that needs to be secreted, it will be carried over to the rough ER. The protein will be threaded in through a hole and pushed in as it's being built and released, and it will fold in the rough ER, and then remember, it will go by vesicle to the Golgi, and then it will be modified and sorted, and then if it's going to be secreted, it will go then by vesicle to the plasma membrane and be released. So now you have really the whole process, at least for protein secretion, from beginning to end. So let's say it's insulin. Let's say the cell that is producing the protein is a pancreas cell. So the insulin gene will be copied into mRNA in the nucleus of each cell in the pancreas. And then the mRNA will come out into the cytosol. Two parts of the ribosome will attach and start making the insulin protein. But there will be a signal at the beginning of the insulin protein that causes the whole ribosome to be moved to the rough ER and attached to the surface of the rough ER. The insulin protein will be threaded into the lumen of the rough ER and released there. And then by vesicle, that insulin will be taken to the Golgi and then eventually out to the released from the cell into the bloodstream. And so that's, an example of the whole process from beginning to some, I guess, to the end. That's the end of it for the cell. Now in the body, the insulin goes and does some other things which are interesting, but it gives you that process. So I'd like to have you see the whole process working, have a concept of that, because none of these concepts that we've learned are fully separate from the others, because everything is working together. There's a lot of connections.